Hey everyone, Dr.Mungli here. In this video, I will be explaining you what is the mechanism of Pcsk9 inhibitors in decreasing LDL cholesterol levels in the blood. So in order to explain the mechanism, I need to give you a brief introduction about the LDL cholesterol synthesis, especially how the cholesterol is synthesized, how LDL is made, what is the role of Pcsk9 and how Pcsk9 inhibitors work here. So as you all know, like liver is the major organ where cholesterol is synthesized. So cholesterol synthesis which is going on in the hepatocyte here, so the liver, hepatocyte and cholesterol biosynthesis is shown here. So the acetyl-CoA is the molecule which is undergoing a metabolism which will make HMG-CoA, hydroxymethyl-glutaryl-CoA, and the HMG-CoA is undergoing a reaction catalyzed by HMG-CoA reductase enzyme to make mevalonate, and that's the rate limiting and regulated step in cholesterol biosynthesis. Now, mevalonate is undergoing a metabolism to convert into cholesterol formation. Now, the cholesterol is the one, whenever we have sufficient cholesterol, so the cholesterol will have a feedback negative effect on HMG-CoA reductase enzyme. And also note that when we have excess cholesterol, so cholesterol is going to regulate a gene that is coding for HMG-CoA reductase enzyme and also the gene that is coding for LDL receptor. If there is more cholesterol in the cell, so there will be negative regulation on LDL receptor expression, so there will be less LDL receptors expressed on the cell membrane. And also note that, so the cholesterol when it is there, so it can be converted into cholesterol ester by ACAT enzyme, acyl cholesterol acyl transferase enzyme. Now the cholesterol ester, it will be loaded on to ApoB100 to make VLDL molecule, and VLDL is secreted into the circulation, and VLDL undergoes metabolism to make LDL molecule. Initially VLDL to IDL, IDL to LDL molecule. Now the 60% of circulating LDL, it will go back to the liver, 40% is taken up by extrahepatic tissues. Now, how LDL is going back to the liver and how LDL is going into the extrahepatic tissue. So LDL to get into the cell, so it has to bind to LDL receptor that is expressed on the cell membrane. Now the LDL receptor has got an extracellular domain and also it has got a cytoplasmic domain. So when LDL is interacting with the LDL receptor, so it is internalized, so it is making an endosome here, so LDL receptor endosome, and eventually it will fuse with the lysosome, and in the lysosome LDL is broken down and the cholesterol gets into the cell. So that's how the cholesterol gets in. Now what happened to the receptor? So in the endosome what happens, the LDL receptor here in the endosome where there is a drop in the pH, and because of the drop in the pH, so there will be change in the conformation of the interaction of LDL receptor with the LDL. So because of the change in the conformation, so LDL receptor will be circulated back to the surface of the hepatocyte membrane or any other cell. So it means there is a recycling of LDL receptor. LDL receptor comes back to the membrane again. Note that LDL is a short-lived molecule. The half-life of LDL receptor half-life is 20 minutes. Every 5 minutes it undergoes recycling process here. So LDL that is binding to the LDL receptor, so receptor is recycled back to the surface, so there is expression of LDL receptor on the surface. It means there is availability of LDL receptor on the surface. Now let's see what happens with the Pcsk9. So this Pcsk9, it is going to control the amount of cholesterol that is present inside the cell. So it means if there is more cholesterol inside the cell, so the Pcsk9 which are synthesized, so they will bind to the extracellular surface here. So the Pcsk9 bind to the extracellular domain of LDL receptor. So LDL binding to the LDL receptor, but there is a binding of Pcsk9 here. So now the entire thing becomes part of the endosome here. Now, because Pcsk9 is bound to the LDL receptor, so the LDL receptor do not undergo conformational change. Because of this, what happens? This entire thing will be getting into the lysosome. Instead of LDL receptors recirculating back to the surface, because Pcsk9 is binding to the surface, to the LDL receptor here, that entire thing which is getting into the endosome So it will go to the lysosome and everything is degraded here. It means LDL receptor is degraded into amino acids. So that means this LDL receptor is not coming back to the surface of hepatocyte to take up more and more LDL molecules. So this is what happens. So PCSK9 molecules, it enhances the degradation of LDL receptor, thereby it decreases the availability of LDL receptor over the surface of plasma membrane. Because of this, what happens? So the circulating LDL is no longer getting into the cell. So what PCSK9 inhibitors does? Now the PCSK9 inhibitors, so most commonly available PCSK9 inhibitors which have been approved by FDA now, there are two of them. One is ulokumab and arilokumab. These are monoclonal antibodies. So these monoclonal antibodies, which are referred as PCSK9 inhibitors. So these PCSK9 inhibitors are monoclonal antibodies against the against PCSK9. So what they will do is, so they are going to bind to PCSK9 molecules. So PCSK9 inhibitors, these are monoclonal antibodies. So they are going to bind with the PCSK9 and thereby they are not going to allow PCSK9 to bind to the LDL receptor. So PCSK9 inhibitors, evolocumab and arilokumab, so they are going to bind with PCSK9, do not allow PCSK9 to bind to LDL receptor. That means LDL receptor which is not bound to the PCSK9, so it can be recycled back to the surface, thereby expression of LDL receptors over the surface is more now. So all PCSK9 inhibitors does is, they are going to make sure that PCSK9 do not bind to the LDL receptor, thereby LDL receptor, once it is internalized along with the LDL, it will come back to the surface. So that recycling of LDL receptor is maintained here, thereby more and more LDL receptors are available over the plasma membrane. That means more and more LDLs can be taken in. So that is what is the role of PCSK9 inhibitors. So since I am explaining PCSK9 inhibitors, let me explain you what is the mechanism of statins on LDL receptor expression. So the statin group of drugs, there are different types of statins. So what they will do is, so they are going to have a competitive negative effect on HMG-CoA reductase enzyme. Statins are competitive inhibitors of HMG-CoA reductase, thereby they are going to decrease cholesterol synthesis. Now when the cholesterol synthesis is decreased, normally this cholesterol, whenever it is increased, it has a negative effect on LDL receptors. Now when there is a decrease in the cholesterol, so this negative effect on LDL receptor is not there. That means more and more LDL receptors are available on the surface. So treatment with statins, it increases expression of LDL receptors. PCSK9 inhibitors, by binding with the PCSK9, it also increases the expression of LDL receptors. So overall, on a larger scale, that means in a bigger picture, so statins increases LDL receptor expression by decreasing cholesterol in the cell. Even PCSK9 inhibitors, by binding to PCSK9, so they will also indirectly increase expression of LDL receptors. So whenever LDL receptors are expressed, that means more and more LDL can be taken into the cell. So this is how PCSK9s work, PCSK9 inhibitors work, and this is how statins they work in decreasing circulating LDL cholesterol. So this is all about mechanism of PCSK9 inhibitors and mechanism of statins in increasing expression of LDL receptor over the hepatocyte membrane, thereby decreasing LDL cholesterol levels in the person who is having hypercholesterolemia. So that's all about it. So thanks for watching. If you have any question, put that question in the comment section below, and I'll see you in my next video. Till then, you take care.